Hello, everyone, and welcome to Lecture 10 in our class on brain and behavior. And this lecture is going to be a bit different. So we've been learning about the structure and function of the nervous system, how neurons work, what their parts are, the different parts of the brain, the peripheral nervous system, and of course we're getting ready for exam 1. And so this lecture won't actually introduce any new content. All it will do is go over what we've seen so far and emphasize the content that's important for the first exam. So I'll talk about a bit about the structure of the exam itself, and then we'll go over some material, what's in, again, focusing on the really important stuff, and the terms and the concepts that you really need to know to be ready for exam 1. So the best thing to do is watch this lecture. You can get the slides from Blackboard. You can even take notes about the important points that I mentioned, and then go back over your notes, go back over the readings, take the old quizzes, even rewatch parts of the old lectures. And so, again, not everything we've covered is going to be on the exam, but a lot of it will be, and this is the important stuff. So this should help you focus. Okay, so the structure of the exam itself, there will be some multiple choice questions. You will only need to write your answers, indicate your answers on the exam itself, so you don't need a Scantron, so don't worry about that. There will be a fair number of fill-in-the-blank questions and matching questions. So the fill-in-the-blank sections will have a word bank, so that is a set of options that you can pick from and fill in the blank with one of those options. Please do use the word bank. The correct option, I guarantee, is in the word bank. Don't come up with something that's not in that list. And matching, pretty standard matching questions. You'll have a list of sort of blanks and a list of options, and you'll match them up. There will be some short answer questions, and these are where you have to write answers in your own words. So you'll have to demonstrate that you understand the concepts. These will often be perhaps a little more open-ended. You'll have to do a little bit more thinking. They will not just be terminological. So I might give you a situation and ask you to explain what would happen in that situation. Maybe I change something about the nervous system, and you have to tell me what the result would be. Or maybe I ask you to explain something, or name a brain structure, and then explain what that structure does. There will be a fair number of diagram questions, and we'll see this for all the exams in this course. That a lot of the content in the course is best presented visually. We're talking about structures, whether it's of neurons, of individual receptors, of the nervous system in general. And so this is best presented visually, and so it's best tested visually. And so I'll give you some diagrams and ask you to fill in labels. It could be labels of brain structures, neural structures. It could be parts of a receptor, for example, but especially that content that we've illustrated diagrammatically, that is by illustration in the course. Those are the prime candidates, especially figures that you've seen in lecture. And again, here you'll have a word bank. You'll have a list of available options. So again, do use that word bank. The answer is in there. And then finally, there will be an essay. And the essay is worth 15% of the points on the exam. And in this case, it will be over the article we've already discussed, the Munoz-Cuevas et al. Article. So go over your notes from that, go over the article itself, and more detail later. But really, it's a pretty short essay. It's not a two-page essay. You could really get it done in a paragraph, maybe a little bit more, just a few sentences. And you'll need to identify what the study is about, what it involves, that is, what the researchers did, what the results are, what they found, and then why it's important. How do we apply the results to our daily lives? So now for the content. So understand how a neuron is built, understand its structure. And we already know that neurons have different components. We have dendrites, we have the cell body, we have the axon, we have the axon terminals. So know what those terms are, know where on the neuron those structures are located, and be able to say what those structures do. It doesn't have to be a lengthy explanation. A phrase or a sentence will probably do it, but understand those different components. Know when it comes to the axon what myelin is and what it does. And also, we talked about different types of neurons. So we talked about things that were classified functionally in terms of what kind of information they convey and where they convey it to. So sensory neurons, motor neurons, interneurons. We also talked about neurons that are classified in terms of structure. So multipolar neurons, bipolar neurons, unipolar neurons. And that's what we have depicted here. And so know what those differences are, be able to identify neurons based on that classification, and why, and know the rule. And the rule, of course, for this group, for this classification system, is how many things are sticking off the cell body. If it's one, it's unipolar. If it's two, it's bipolar. If it's more than that, it's multipolar. So pretty simple, but be able to apply that and explain it. And of course, we talked about how our nervous systems are composed of more than just neurons. There are also glia, or glial cells, or neuroglia. I usually just stick to glia, or glial cells. So for each of these, again, the point is not to write a lengthy description of what each one does, but a phrase, a sentence would be sufficient. So know what astrocytes are. Know what they do. They have a couple different functions. So be able to say a couple phrases about what astrocytes do. Microglia, a different kind of glial cell. So know what those do. Ependymal cells, those are those cells that lie in the interior of the ventricles of the brain. So be able to describe their function. In this case, they produce cerebrospinal fluid. So know that, be able to say it. And then, of course, we have those cells that produce myelin. And we have different myelin-producing cells in the central nervous system and the peripheral nervous system. So in the central nervous system, we have oligodendrocytes, and those produce myelin, but they can produce myelin for multiple axons. And that's different from Schwann cells. Schwann cells also produce myelin, but they're found in the peripheral nervous system, and they only encapsulate one axon. And we also know from our unit on the repair of the nervous system that that is an important difference. The kind of myelin, the content of the myelin differs between oligodendrocytes and Schwann cells. And that accounts for the different abilities of the central and peripheral nervous systems to heal themselves. So understand the differences between the glial cells, but also be able to describe the function of each type. For the brain itself, the structure of the central nervous system. Know what the four lobes are, know where they're located, and then also know what each one does. Again, a sentence will suffice here. Maybe a couple of functions for each one. So this is a diagram from your book. It's a good diagram. And this is, for those diagram questions, this is the kind of diagram that you might see. So perhaps it's this picture, but with the words all removed. You don't need to know all the structures on this diagram. Know what the lobes are. Know what each one does. And this is not one of the lobes, but it's depicted here, so I'll mention it. Understand the cerebellum. Know what the cerebellum does. Be able to identify it in a picture. Know your anatomical terms. So not just the structures of the brain that I'll mention, although those are important, but we talked about those anatomical planes. The horizontal plane, the sagittal plane, the coronal plane. So know what those are. Be able to identify a picture of the brain if it's one of those slices or planes. Know your directions as well. So know the difference between anterior and posterior, ventral versus dorsal, lateral versus medial, superior, inferior, don't worry about as much. Rostral and caudal is on this diagram from your book. I don't use that term much at all, so don't worry about that. But the other ones, anterior, posterior, ventral, dorsal, medial, lateral, you should know those. And it's not just the lobes of the brain that are important. So this is another diagram that we studied in class. And the point here is certainly not to memorize all of these structures because we haven't covered all of them yet. But I'll highlight a few important ones. First, there's the thalamus, which, as we know, is our sensory routing center. So all of our senses, except for our sense of smell, send information through the thalamus. It's processed a bit in the thalamus, but then it's sent on to the other parts of the cerebral cortex that process that particular kind of sense. So your sense of touch information first goes through the thalamus, and then is sent to somatosensory cortex. Visual information goes to the thalamus, and then goes to visual cortex and the occipital lobe. So know that that's what the thalamus does. The corpus callosum is that bridge, that band of white matter that connects the two hemispheres, the left and right hemispheres. And while you can survive without one, if the corpus callosum is cut or doesn't develop, your two hemispheres are pretty much independent. So know what the corpus callosum is, be able to identify it on a diagram like this one, or even a picture of the brain that's on an anatomical plane. This is the sagittal plane. The cerebellum I've already mentioned, so be able to identify it, know what it does. It's involved in balance coordination. And the brainstem. You don't need to know the individual parts of the brainstem, but know that the brainstem, first where it is, but also that it sustains your vital functions. Okay, so beyond just the lobes and parts of the brain we just went over, know those brain systems, those sort of networks of regions that work together. And the point here is not to memorize the individual parts of these systems. There are a couple structures that are particularly important. But the basal ganglia, know what the basal ganglia do, know roughly where they're located. So they're involved in selecting actions and processing rewards. So that's important to know. And the limbic system, as I've already mentioned in class, whether it deserves the label of system or not is debatable. In fact, it probably doesn't. But the structures within it, some of them are very important, and some of them are structures that we're going to come back to again and again throughout the course. So those are the amygdala and the hippocampus. So the amygdala is involved in emotional processing, particularly fear. The hippocampus is involved in memory, different kinds of memory, and we haven't had our unit on learning and memory yet. So we haven't gone over all the different types of memory. And the hippocampus isn't required for all of them. But it's required for remembering facts, remembering events in your life, remembering locations, so spatial navigation. So the hippocampus is important for forms of memory like that. So know that. Know where the hippocampus is, where the amygdala is. They're both located on the underside of the temporal lobe. The rest of the limbic structures, don't worry about for now. So this is a diagram from your book. This is where those structures are. And again, this is a cartoon version, so it could be possible that it is a diagram, it forms the basis of a diagram question. But also anatomically, we identified the amygdala and the hippocampus on actual images of the brain. So keep that in mind as well. And there are more structures. So as I've already mentioned most of these. So the basal ganglia, the thalamus, the difference between gray matter and white matter, the ventricles, the corpus callosum, again, the amygdala and the hippocampus. These are all structures that we've talked about a fair amount. So be comfortable identifying them in cartoon form, in the pictures we've seen that are actual images of brain slices. So get familiar with those. And again, the reason we're emphasizing that is that these come up again and again throughout the course. So it's important to know them now. The peripheral nervous system. So we talked a bit about the peripheral nervous system, and I've already mentioned that the cranial nerves, it's not like you have to know the individual numbers and functions of each of the cranial nerves, but be able to compare the peripheral nervous system to the central nervous system. So we know, for example, we've already talked about how the CNS is not very good at repairing itself compared to the peripheral nervous system. But the CNS also has forms of protection that the PNS doesn't. So the CNS has astrocytes and microglia. The PNS does not. The central nervous system has meninges, the dura mater, the arachnoid, and the pia mater. And those are those membranes that cover and protect the central nervous system. So the CNS has those. The PNS does not. And those form sort of the boundary between the CNS and the PNS, because the peripheral nervous system, those nerves will exit from the spinal cord, and they're no longer protected by the meninges. And so that the meninges form sort of the border between the CNS and the PNS. For the peripheral nervous system, understand the division between the sympathetic and the parasympathetic nervous systems. Know what those two do. So we've used the rhyming mnemonic devices, fight or flight and rest and digest. Be able to get a little more detailed than that. So know individually how individual components of our physiology are affected by the sympathetic and parasympathetic nervous systems. So heart rate, for example, the diameter of airways, digestion. How are these affected by the sympathetic and parasympathetic systems, and what is the purpose of those systems? What do they let us do? For reflexes, again, how we label neurons. We have sensory neurons, we have interneurons, we have motor neurons, and obviously those relate to the different kinds and structures of neurons we've seen. So sensory neurons are unipolar neurons. Inhibitory neurons, the interneurons, and the motor neurons are multipolar. But understand those physical distinctions and those labels. So we have sensory neurons that carry information from, in the case of the D-jerk reflex, the muscles, but it could be any component of the sensory system, carries that information to the spinal cord. The motor neurons do the opposite. They carry information from the spinal cord out to the muscles. And so they're what makes a muscle contract as part of a reflex. But obviously the sensory neurons are the first step. They have to detect the environmental stimulus. So we have sensory neurons, motor neurons, and those interneurons that relay information from one neuron to another. They don't connect to muscles. They don't connect to sensory organs. They just communicate between neurons. But they're an important part of even something simple like a reflex. They're a very critical part of the central nervous system. Your brain is almost entirely interneurons. So know what that term means and know why it's important. Some other terms, afferent. So those are, in this case, the sensory neurons. But afferent describes any neuron that brings information to the central nervous system. So it carries it to the spinal cord, or it could even carry it directly to the brain. So it's a description of information flow. And of course, there's the term efferent, sometimes pronounced efferent. And those are neurons that bring information away from the central nervous system. In the case of a reflex, it's bringing information from the spinal cord to a muscle. It is the motor neurons. But again, efferent describes any neuron that takes information from the central nervous system and puts it out somewhere in the body. The physiology of neurons, and this is often the toughest content for this exam, and perhaps even in the whole course. So know the ions that we've talked about so far. For action potentials, it is really all about sodium and potassium. For synaptic transmission, we brought in a couple of other ions. Calcium is an important one. And when it comes to inhibitory signaling, chloride is an important one. So know those four ions, know their charges, and the point there is not to just memorize a bunch of positive and negative signs. It's to understand how those charges and how movement of these ions affects the physiology of the neuron. How does it affect membrane potential? How does it affect action potentials or synaptic transmission? So know where these ions tend to be concentrated. Sodium tends to be outside the neuron. Potassium tends to be inside. Chloride and calcium both tend to be outside. And so that's important because it's what allows our synaptic transmission and action potentials to work. You have to have those concentration gradients, or the ions won't move when those channels open. So again, know the ions, know where they're prevalent, know their charges. And we have things like channels that allow sodium, potassium, chloride, calcium to move across the membrane. Otherwise, those ions are stuck. And we have those sodium-potassium pumps, which are those little molecules that use energy to maintain those concentration gradients. So they pump sodium out and pump potassium in, which is not where those ions want to be in terms of equilibrium. Sodium obviously wants to go into the neuron, potassium wants to go out, and that is exactly what happens when an action potential occurs. But the pumps are there to maintain those concentration gradients. So know about channels, what it is that they do, understand what pumps do, and again, understand ions, what their charges are, where they're prevalent. and the related terms like membrane potential, so the difference in charge between the inside and the outside. Know that the outside is usually more positive than the inside. So understand what the term membrane potential means. And then also, not just in terms of memorizing the definition, but understand what ions do to the membrane potential when they move across that membrane. And this is important for our action potential unit. It's important for our unit on synaptic transmission. So when sodium comes pouring in during an action potential, it raises the membrane potential. We have that upward part of the spike. When potassium leaves, it causes a drop. Positive charge leaving the neuron lowers the membrane potential. Chloride in an inhibitory synapse, usually the neurotransmitter GABA, will activate receptors, and the corresponding ion channel will let chloride in. And if we think about sodium, sodium is positive. If it goes into the neuron, it's going to raise the membrane potential. Chloride is negative. If it goes into the neuron, it's going to have the opposite effect. It's going to drop the membrane potential. It's going to hyperpolarize that neuron. And so think about what each kind of charge moving across the membrane does to the membrane potential. Because that's important, again, for understanding the action potential. It's important for understanding synaptic transmission, and in particular, understanding the difference between excitation and inhibition when it comes to synaptic transmission. So an excitatory synapse, when, for example, glutamate binds to a receptor and lets ions in, the ion it usually lets in is sodium. And so sodium comes pouring in, and it depolarizes the neuron. It brings it perhaps up to threshold where it can fire an action potential. But sodium coming into the neuron always depolarizes it, always brings that membrane potential up. So understand how that works and how the movement of charge across the membrane relates to ion channels and how it relates to the membrane potential. And when it comes to those ion channels, know what voltage gating is. So voltage-gated channels are sensitive to the membrane potential. When you depolarize a neuron enough, those channels will open. For sodium channels, it only takes a little bit of deviation from resting potential. You only have to go up, call it 20 millivolts. For those potassium channels, it's the top of the spike. So it has to be positive 40 millivolts. So it has to move the membrane potential a lot before those potassium channels open, but they are still voltage gated. So know what voltage gating is and how it works. The action potential. So we went over this a fair amount, and I know that this is one of the hardest components of the course, but it is critical to understand this not just, again, as a series of definitions, but as a process, as a dynamic process. It's a series of interlocking parts, so to speak. So know the various phases of the action potential. Resting potential, around minus 60, minus 69, minus 70 millivolts. The neuron is sort of just sitting there. That sodium-potassium pump is working to maintain those concentration gradients. But it takes a stimulus, whether from an electrode, if we're doing it artificially, or from an excitatory impulse on the dendrites. That is enough to raise that neuron's membrane potential to threshold. And that threshold is the point at which those voltage-gated sodium channels will open. And once those open, sodium comes pouring in very quickly. That's the upward swing of the spike of the action potential. Once you get toward the top of that spike, the membrane potential is so high that those potassium channels that are voltage-gated, those start to open. And now potassium goes rushing out. So it goes up because of sodium. It comes crashing down because of potassium. And then the sodium-potassium pump works to reestablish those concentration gradients and bring the neuron back to resting potential. So know how that process works. Know why the membrane potential moves the way it does and how it relates to the movement of ions. Know the timing of those ion channels opening, closing, and activating. So understand that process. And one of the best ways to test your knowledge of your understanding of that process is thinking about what happens if something goes wrong. So we talked about some toxins that work on ion channels or components of the membrane of neurons that alter action potentials or prevent them entirely. So this is the pufferfish on the left, this is an algal bloom on the right, but these things both produce toxins that block sodium channels. And we talked about what that does to an action potential. So think about what happens if you block sodium channels. What does that do to the action potential? And in this case, you can't have an action potential. If you go up to threshold, the sodium channels are blocked; they can't open. So even if you go up to their usual threshold, they can't do anything. So you don't get that upward swing of the spike, which means you don't get that downward swing of the potassium channels open. They never get up to their threshold. Similarly, what happens if potassium channels stop working? So this is a black mamba and a cane spider, but their venoms block potassium channels. So the first part of the action potential works. You get sodium channels opening if you reach threshold because they haven't been blocked. They're fine. But think about what happens if you block those potassium channels. What will the action potential look like then? What happens if those pumps stop working? What happens if if they stop right when an action potential happens? What would that action potential look like? What happens if they haven't been working for a long time? So those sodium-potassium pumps are what maintain those concentration gradients. Think about what happens if those concentration gradients don't exist. If those pumps aren't working, those ions will eventually reach their equilibrium. They'll get to where they want to go. It just takes a really long time. But without that pump working to maintain those gradients will eventually go away. So think about that process. And again, that's a really good test of how you understand the dynamics of the action potential, because it's not something you can memorize. There are lots of ways for these things to be disrupted. And again, you could memorize the sequence of events in the action potential, and it's good to know them. But if you understand this as a dynamic process, as a series of interlocking parts, then you can predict what will happen if this system is disrupted in a very specific way. So that's important too. So synaptic transmission. We know how the action potential works. We also need to know how synaptic transmission works. And one important distinction here is that between the presynaptic and the postsynaptic neurons. So know what those differences are, know what those terms mean, and know the different processes that happen in the presynaptic and postsynaptic neurons when it comes to synaptic transmission. More on that in a second. Know what synaptic transmission does on the large scale. So we can have an excitatory postsynaptic potential. We can have excitation of the postsynaptic neuron. And that usually happens by, glutamate is the most common neurotransmitter, but many neurotransmitters will excite the postsynaptic neuron, make it more likely to fire an action potential through an excitatory postsynaptic potential. And as I've mentioned, that is typically done by letting sodium into the dendrites. So that's going to depolarize the membrane potential of that postsynaptic neuron. It makes an action potential more likely. You have the opposite as well. You have inhibitory postsynaptic potentials, IPSPs. And this is often done with the neurotransmitter GABA, or inhibitory neurotransmitter. And an IPSP happens when an ion channel opens and lets in chloride. So chloride is a negative ion. When it comes into the dendrite, it hyperpolarizes the membrane potential. So the membrane potential is usually sitting at -60 or -65, -70. And if you let in negative charge, that pushes the membrane potential even farther away from zero. It hyperpolarizes the membrane potential, hyperpolarizes the neuron, and makes it less likely that an action potential will occur. You'd have to have that many more EPSPs to get up to threshold. So understand that difference. Understand the difference between EPSPs and IPSPs in terms of the results, making action potentials more or less likely, in terms of their mechanism, letting in sodium versus letting in chloride, and in terms of neurotransmitters. EPSPs most commonly glutamate, IPSPs most commonly GABA. And again, relate that to the ions we talked about, sodium and chloride in this case. And understand the mechanics of synaptic transmission. On the presynaptic side, we have an action potential coming into the axon terminal. We have calcium channels that are voltage gated that sense that action potential, open up, let in calcium. Calcium makes those synaptic vesicles, which are full of neurotransmitter, makes the vesicles bind to that presynaptic membrane, releases the contents, releases neurotransmitter into the cleft, and then that neurotransmitter can diffuse across the cleft and bind to receptors on the postsynaptic side. So that's part of what happens after vesicle release. You have diffusion of neurotransmitter across the cleft, binding to receptors, but also know what happens to neurotransmitter afterwards. We don't want neurotransmitter just sitting there forever in the cleft, so it either gets chewed up by enzymes or it gets reabsorbed into the presynaptic neuron by the process of reuptake. So know those terms and understand, again, that sequence of events. Understand what binding to receptors on the postsynaptic side does. More on that in a moment. But understand how we get from an action potential to the release of neurotransmitter into the cleft, and know what happens to neurotransmitter after it's released. And when it comes to neurotransmitters, we need to know the ones we discussed in class. Again, you don't need to know an exhaustive list. You don't have to write an essay about each one. Know what they do. Know their basic function. So we talked about a handful of them. And again, a paragraph is not necessary. If you can name a couple of functions, a couple of phrases, that is sufficient. So know what they do in the central nervous system. We also talked about some neurotransmitters in the peripheral nervous system having a slightly different role. So for example, acetylcholine in the central nervous system, it's involved in memory. In the peripheral nervous system, it's involved in the contraction of muscles. Most of these neurotransmitters we just talked about in the context of the central nervous system. But norepinephrine is another example. Norepinephrine involved in alertness. Also, it's the neurotransmitter of the sympathetic nervous system, part of the PNS. So we have glutamate, we have GABA. Those are our excitatory and inhibitory neurotransmitters. We have acetylcholine, dopamine, norepinephrine, serotonin. So know what those neurotransmitters do. Know their prevalence, and that's not as important for acetylcholine, dopamine, norepinephrine, serotonin. There aren't that many of those neurons, relatively speaking. We're talking about tens of thousands. So know that rough number. You don't need to know between dopamine and norepinephrine, which is more common. But know that glutamate is our most common neurotransmitter. Know that GABA is our second most common one. It's our inhibitory neurotransmitter. And the others are all much less common than those first two. And also know where they're made. So glutamate and GABA are made throughout the brain. But acetylcholine, dopamine, norepinephrine, serotonin, there aren't that many of those neurons. And those neurotransmitters, those neurons, originate in some evolutionarily old structures. So know those structures for those particular neurotransmitters. Also, as a side note, again, as we've mentioned, most of these neurotransmitters only go by one name. Serotonin goes by 5-HT as well, so know that synonym because that's going to come up again and again in the course. We'll see the term 5-HT referred to over and over. So it's important to know that's a synonym of serotonin. Postsynaptic side of synaptic transmission. So know your receptors. So first know what a receptor is. So it's a protein that a neurotransmitter binds to. That's the most basic definition. And we know that they come in two basic flavors, metabotropic and ionotropic. So know what that difference means, knows what those terms mean, and we need to know what that means structurally. So an ionotropic receptor has an ion channel that's part of the receptor. It's built into it. A metabotropic receptor has a part that the neurotransmitter binds to, but then it sends a chemical signal over to ion channels so that they can open. That process takes longer, but it's also important to know that those ion channels are not physically part of that receptor. The receptor can have lots of effects because it uses this indirect signal that can go lots of places. An ionotropic receptor, all it can do is open. A neurotransmitter binds to it, the channel opens, it lets some ion through, but a metabotropic receptor, a neurotransmitter binds to it, and it sends off a chemical signal that can do any number of things, including letting distant ion channels open up. So in terms of function, what that means is that ion channels tend to be very fast. So the ion channel is right there. It's part of the receptor. So when that neurotransmitter binds, that ion channel immediately opens up. So ionotropic channels respond very quickly. Metabotropic channels tend to be slower. They have to use this indirect signal that takes a while, relatively speaking. Also, the flip side of that is that ionotropic channels, their effects tend to not last very long. They open up, let ions through, but then they close again. And those concentration gradients are established pretty quickly afterwards. Metabotropic channels, those signals they send are indirect, so they take longer. But those signals can go as far as the cell body and the nucleus. You can change which genes are active. And those can be very long-term effects. So know that difference. So between metabotropic and ionotropic channels, how they're different structurally, but also how they're different in terms of their effects, the length of time it takes for them to take effect, and how long those effects last. And then finally, as I mentioned, the essay on the exam will be based on the Munoz-Cuevas et al. Paper that we discussed in class. And so some basic questions. You have to know what they studied. So they're looking at dendritic spines in mice. And so they're looking at the effect of cocaine on those dendritic spines. Does it make those dendritic spines go away? Does it make them appear? What is the relative rate of those two processes? So you have to know what it is they're looking at, what they care about. And also, of course, they're looking at behavior, as we'll see. And know what they did experimentally. Know the basics of that experiment. And we know they did a couple different experiments that are all related. They all involve the administration of either cocaine or saline. And we know that there was one experiment that looked at this in a long-term setup, so imaging every 48 hours to look for dendritic spines, and then also measuring how much they move around after cocaine administration. We know there was another experiment looking at the short-term effects. We know there was another experiment looking at the behavioral effects. So be able to describe each of those different parts of the study in terms of what they did, what it is they're looking at, what were the results. So we know that cocaine administration increased the rate at which new spines were grown, didn't increase the rate at which they were lost, so those spines stuck around. We know that it wasn't related to the mice moving around more, even though that's something that cocaine does. And we know that it's related to that cocaine condition place preference test. So they tested which space of those two chambers was preferred by the mice, and then gave half of them cocaine, and measured to see what their preference, how much their preference changed. And then related that back to how much their brain structure changed. So understand those results, know those terms like locomotion, cocaine condition, place preference. So know those important terms, know what the results were, and be able to describe them. And again, this is not going to be a long essay. You can get this done in a paragraph, but you could even practice writing these essay questions. And I'm laying out what the question is going to be about. So you can practice writing that essay. And then what's the interpretation? What do the findings mean? We talked about that in class as well. So what is it that these findings tell us? Obviously, we don't really care that much about what cocaine does to the brain of a mouse, what cocaine does to the behavior of a mouse, but we care about what it does to people. And so what do these findings tell us about things like addiction, about drug-seeking behaviors? So again, you could practice writing that, but be prepared to discuss that article in a short essay. Okay, and with that, I will say good luck. That is, again, an idea of what you should really focus on as you start to study for this exam. So we have the structure and function of the nervous system. We have the Munoz-Cuevas et al. Paper. We have action potentials and synaptic transmission as dynamic processes. And I've already gone over the structure of the exam. But we're going to ask these questions in a number of ways. Some are visual, some are short answer, some are matching, some are multiple choice. So some people find flashcards useful for the terms. That could be a good approach for you. And again, you can practice writing out those essays, thinking about what happens if you disrupt one of the steps in synaptic transmission or one of the steps in the action potential. And again, the point there is to think about this not as a sequence of steps to be memorized, although you should know the steps, but as a process with interlocking parts. So if you go over this lecture, if you go over those slides that are posted, that should really help you focus and prepare for the exam. So with that, I will say good luck, and I will see you in class.